So far we have seen two kinds of maps in the primary visual cortex, the orientation maps and the ocular dominance maps. In the orientation maps we have seen that the orientation changes continuously as a function of cortical location. So, as you probe the visual cortex along a line at various points, you will see that the orientation changes over the full cycle from 0 to 180 degrees, and it is periodic. So, therefore, once you reach 180 degrees you start all over again from 0 and you progress towards 180 degrees again. And so, although the orientation changes continuously at most points, at certain points at isolated points you do not have this kind of a continuous variation, and such points are called singularities. So, let us see how they look like. So, for example, here the orientation changes continuously from yellow to yellow to green. So, that is yellow is 45 degrees and green is vertical 90 degrees, but at certain points so lots of colors meet at certain points. So, these are those points. These points are mathematically called singularities, and in visual science they are called pinwheels, because they look like wheels with spokes around them. So, at this point neurons respond to all sorts of angles. So, because at this point in the neighborhood you have red here which is horizontal lines, you have yellow here which is 45 degrees and green which is vertical, and so on and so forth. And again you have two kinds of pinwheels if you look at it here. So, there are two kinds of singularities or two kinds of pinwheels. So, for example, at this point you go from red, if you go in the anti-clock direction, you go from red to yellow to green to blue and back to red. But whereas if you look at some other point, if you take this point, you go from clockwise direction, you go from red, yellow, green, blue. Okay, so you encounter the same sequence of orientations. At some points you encounter some sequence of orientation when you go in the clockwise direction. At other points you encounter the same sequence when you go in the anti-clockwise direction. Okay, so these are the two kinds of pinwheels, and it turns out that both occur in about the same frequency. Okay, and in addition to the pinwheels, there are also these line-like regions, what are called fractures, across which orientation changes drastically. So, if you go back to this plot, right, some places, like I said, it varies continuously, but some places it varies drastically. You can see a kind of a line which separates two orientations. So, look at that. You have very sharp transition from purple to red. Look at that, the sharp transition from blue to yellow. So, okay, so you have continuous stretches, then you have sharp fractures where across a line there is sharp change in orientation. Then you have singularities of pinwheels, where around a point in the neighborhood of a point there is a discontinuity, and there again you have two kinds of pinwheels, right, as we have just described before. Now, if you look at ocular dominance also changes continuously as a function of cortical location, and they are organized into parallel strips, right, which sometimes branch and terminate. So, if you look at this map, this ocular dominance map, so they are organized as parallel strips, right. You have this, so you have black and white parallel strips if you look at a small patch of the ocular dominance map, but sometimes these strips branch out like you see here, right, and also terminate. So, you see this little stretch terminating here, or its branch terminating here, and so on. So, these are some of the properties of ocular dominance maps. But the thing is, both kinds of maps share the same space, same cortical space in the primary visual cortex, right. Now, the question is, how are these two maps related? Because if you overlay these two maps one on top of the other, right, how are these two maps related? So, here there are two principles which people have observed to describe the relationship between these two maps. One is that iso-orientation slabs often cross the borders of the ocular dominance bands at approximately 90 degrees. Okay, so, in this map where they have drawn these lines to indicate both maps in the same space. So, these white lines indicate the borders of ocular dominance maps, and the colored patches, right, as we have seen before, indicate the orientation maps. Now, what the first principle says is iso-orientation slabs often cross the borders of the ocular dominance bands at approximately 90 degrees. So, these are the borders of the ocular dominance maps, and and for example, look at this spot. So, there is a sharp change in orientation, this iso-orientation line, and the border crosses this line at approximately 90 degrees. So, here it's not a great example, but if you look at this point, right. So, the ocular dominance border right crosses the iso-orientation map at approximately 90 degrees. So, look at that. Okay. So, you can find a lot of such examples. Then the singularities tend to align with the center of the ocular dominance bands. So, so these are the ocular dominance bands. So, if you look at two approximately parallel white lines, right, the stretches of the map between them are the ocular dominance bands, and so the pinwheels often occur roughly in the center of these bands. So, let us look at some examples. So, here is a pinwheel, right, here is a pinwheel, it occurs roughly in the center, right, of this band. So, again here is a pinwheel. Well, let us look at a better example here. So, here is a pinwheel, it roughly occurs right in the center of this band. So, here is another one occurs in the center. So, okay, so in lot of with high frequency, right, these singularities tend to align with the center of the ocular dominance bands. Okay, so we have seen different kinds of maps from the visual system, from somatosensory system, and so on. Now, the question is, we just described them as they are found in real brains, and we also noticed that they are dynamic, because in case of ocular dominance maps, we have seen an example, right, how the map changes when you change the stimulus, right, when the eye is sutured, the neurons responding to the open eye or intact eye were more in number than neurons which responded to the sutured or the closed eye. So, they are also dynamic. Okay. So, now these are just observations, but what are the mechanisms, what are the underlying principles by which these maps form? Okay, because we have seen that the maps are not hardwired, they form by a process which you can describe as self organization. The maps are dynamic and they adapt to changing conditions of stimulus. If you change stimulus, the maps change. What are these mechanisms? So, let us take an illustration, right. So, first of all, we know that since there are neurons which respond to certain oriented bar, right, we have seen that a given neuron responds the highest when it is shown a certain bar with certain orientation, but if you change the orientation, neuron responds lesser and lesser. So, that means if you plot the response of the cell as a function of the orientation, it will look something like this, like this bell curve, right. High response is highest for certain orientation, and as orientation changes from that optimal point, response decreases. So, so now let us introduce the rule 1 of map formation. So, let us assume that there is a neuron which responds to a stimulus like this, a vertically oriented bar, right, and so if you present this stimulus to this neuron, it does not give great response, and so this is the response produced by the neuron. Okay. It is tuned to this orientation, therefore, if you present this vertical bar as a stimulus, it produces a small response, right, but then you present the same vertical bar again and again to the neuron, right, and so on repeated presentation, right, the same vertical bar will be successful in eliciting a stronger response in this neuron after some presentations. That means, at the end of that, the tuning of the neuron changes from its original angle. This looks something like 70 or 80 degrees, right. From this angle, the orientation will probably change very close to the orientation of the stimulus, which is 90 degrees. So, the tuning also changes to approximately to 90 degrees, right. So, on repeated presentation of a stimulus to a given neuron, the response of the neuron is likely to change in a way that the neuron gets tuned to the stimulus. Now, let us look at rule 2. So, rule 1 talks about a given neuron and its relationship to the stimulus and how its response changes to a stimulus. Rule 2 talks about how the neighbor of a neuron, right, responds to a given stimulus. So, in this example, let us assume that initially I give a stimulus which is like a 70 degrees oriented bar, and neuron A happens to have a tuning which is exactly equal to that or very close to that stimulus, but then A's neighbor, right, happens to have very different orientation initially. So, this one responds to a horizontal bar. Now, on repeated presentation of this stimulus, so your 80 degree or 70 degree stimulus, right, neuron A does not change much, and because anyway it is already tuned to that orientation, but neuron A now starts moving towards 80 degrees. So, this is 0 degrees. So, let us call this 70 degrees, right, and so from 0 it might, so it gradually turns, right, towards and becomes maybe 60 degrees. So, tending towards, right, 70 degrees. Okay. So, neuron A is now slowly changing its tuning towards that of the its neighbor, because neighbor is already tuned towards 70 degrees. So, if you combine these rules, right, in a two-dimensional sheet of neurons, right, and we can actually implement these neurons and using mathematical equations and simulate them on a computer, right. Let us see what happens if we do that, and that kind of a map model is called a self-organizing map or a SOM, right, and a lot of interesting map phenomena in the brain can be explained even using simple mathematical models like this. So, in a self-organizing map, it consists of a two-dimensional array of neurons like what you see in this slide, and then you can present your stimulus in the form of a vector, right. Your stimulus is here. So, that could be like your stimulus could represent maybe a vertical bar, or if it is a visual stimulus, or a frequency if it is an auditory stimulus, and so on. Now, let us assume that a given stimulus produces a lot of response in a certain neuron, a high response in a certain neuron. So, then if you present the stimulus repeatedly, right. So, let us assume that this neuron is initially only like at 85 degrees. On repeated presentation, right, it gradually tends towards 90 degrees. Okay. And so, initially let us assume that the neighbors of this neuron are at a very different angle, say 45 degrees, but on repeated presentation the neighbors also tend towards 90 degrees or maybe 85 degrees. So, basically neurons tend to move towards stimulus in flocks, right. If a central neuron moves towards a stimulus, right, or its input connection pattern moves towards stimulus, right, the connection patterns of the neighbors also are dragged in the same direction. So, that nearby neurons tend to respond to similar stimuli. So, that is how you get this kind of a continuous map structure. So, you can see that in this simple animation. So, here I have a two-dimensional array of neurons. So, what you are seeing here is, so you have a bunch of dots which represent the stimuli, and the stimuli are two-dimensional stimuli. These are vectors in 2 dimensions. So, the stimuli are clustered in certain locations. So, you have lots of clusters. Okay. So, you have one here and so on and so forth. So, you see that wherever there are clusters, input stimuli occurring in clusters, right, the neurons in the map tend to respond to those clusters, and lot of neurons are clustered or moved towards points in the input space where there is a high density of points. So, these kinds of maps can be used to explain or organize information in the artificial world, in the world of engineering. So, for example, in this simple example, they have constructed a map of world's poverty. Okay. So, this map gives an idea of the poverty levels of different countries of the world. So, they have taken 39 indicators of poverty from various nations and trained a map, and the map that they got by this training looked something like this. So, you see that some of the poorest countries are colored in the purple, okay, in this corner of the map. Some of the wealthiest nations are colored in yellow, and then this corner, right. Countries with intermediate levels of poverty are somewhere in between, right, and so on. Okay. So, all this happens automatically without any specific hand coding, or you do not have to manipulate the map, right. All that you have to take is take a bunch of properties that indicate the poverty level. These are the indicators, and you train the map. So, if you now once the map is trained, you can superimpose the map on the real world map, and you can apply the same color to the various countries on the map, and you can see where there are wealthiest countries, where there are poorest countries, and so on. So, this part of the map represents, right, some of the poorer nations of African continent, right, and then so this part of the map indicates some of the nations of the developed world, US, Canada, and Australia, right, and so on and so forth. Okay. So, you can apply the maps to organizing information in the real world, in the artificial domain in many ways. So, now let us look at how you can use the same kind of a map algorithms, right, to explain maps in the brain. So, let us take the first example, and this example is from the auditory cortex of the bats. Now, bats are interesting, they are blind visually, but they use sounds to navigate, right. In fact, they throw sounds out into the world, and based on the echoes that they get from the world, they navigate. So, this is and find out where various objects are. So, this process is called echolocation, right. So, this is called echolocation, and so these sounds are ultrasonic waves. So, the normal human range of sounds is somewhere between 20 hertz to 20 kilohertz, whereas bats operate around 60 kilohertz. Okay, and so when they send these sounds out as impulses, right, from the time taken for the signal to come back after it hits a target, right, you can find out, the bat finds out the distance of the object from itself, and also from the change in frequency that occurs after it bounces off a moving target, right, it finds out the relative velocity of the target with respect to itself using the familiar phenomenon of Doppler's effect. So and so on. So bats brains, right, perform very complex signal processing operations on the signals that the return signals that they receive, right, from the world, and navigate through the three-dimensional world. Now one so there are lots of complex maps inside the bat's brain which have been explored by many neurobiologists, right. One of the simplest aspects of it is there is a part of bat's auditory cortex, right, where there is a simple map of the input frequency along a kind of a linear stretch of the auditory cortex. So it is almost this part of the bat's brain is performing some kind of a Fourier transform, okay, an FFT of the input frequencies, that is you have lowest frequencies, right, processed in one end of the map, and highest frequencies processed in the other end of the map with a continuous variation frequency along the length of the stretch of the cortex. So this phenomenon has been simulated in this study by Martinez et al. Right, using the same self-organizing map model. So they have taken an array of neurons of size 125 by 25, and initially each neuron is tuned to certain frequency. Initially the tuning frequency is random, so it looks something like the picture on the left side, right. So nearby neurons in this map do not respond to similar frequencies in this map, because so you can see the colors are all jumbled. But whereas as you train the map using a bunch of frequencies from around the frequency of interest for the map, that is around a band around 60 kilohertz, right, the map self organizes, and you see there is a gradual transition of frequencies from one end of the map to the other, just like what you find in the bat's auditory cortex. Okay, so that is a fairly simple phenomenon. Let us look at a slightly more interesting, slightly more complex phenomenon. This is from the domain of somatosensory cortex or touch. So we have seen these maps before, right. And on the left you see the somatosensory cortex in humans and the somatosensory map in humans, and on the right you see the motor map. Okay, so these kinds of maps have also been explored in monkeys. So in this experiment by Mersenich and colleagues, they have taken a monkey and mapped the fingers of the monkey, right, and you see in this schematic the maps of different fingers of the monkey. So one is its its index finger, and two corresponds to its middle finger, and so on and so forth. Okay, so this is the part of the somatosensory cortex of the monkey, right, and which responds to the corresponding finger of the monkey's hand. So once the fingers are mapped, they amputated the middle finger. So this finger is amputated, and therefore after amputation the neurons which were earlier responding to the middle finger, right, all these neurons they have nothing to respond to. So initially they remain silent because finger is amputated. But after several weeks something interesting happened. So these neurons were initially silent because the finger is missing. These neurons slowly started to respond to the neighboring fingers. So for example, the area 2, right, which corresponded to index finger, right, now started responding to the middle finger partly. Neurons of the region 3 or the middle finger, which were earlier responding to the middle finger, now began to respond partly to the index finger, that is area, this is finger 2, and partly to the ring finger, which is finger 4. Okay, so it is like what happens imagine what happens in a big company when a whole division is fired. All the people in a big division is fired, they are fired. So these people now maybe are relocated or to different other divisions of the company, so that they do not lose their job. So that is what is happening here. The area 3 neurons which lost their job in a sense, right, which cannot respond to the finger, now part of them are now doing the job of responding to finger 2, and the rest are responding to finger 4. So this phenomenon can be simulated very easily using a self-organizing map model. So this was done in a study by Ritter, Martinez and Schulten, described in this reference here. So, they have taken a simple model of the hand which is shown here. Basically, it is a simple two dimensional geometric pattern where you have the fingers. So, this is the thumb, this is the index finger, middle finger, ring finger, and there is no pinky, the little finger is omitted in this simulation, and this is the palm, this whole region is the palm. And they trained the map on this distribution of points, and they found that certain parts of the map respond to certain parts of the hand. So, this big region corresponds to the palm, and so this corresponds to the thumb, and this corresponds to the ring finger, and this is the middle finger, and so on and so forth. So, once the map is trained right for the normal hand, then they amputated the map, which means that they basically removed all these points from the training data set and then trained the map further. So, when they did that, what happened is, so these neurons initially have nothing to respond to, but after some training the same neurons in the same location now began to respond to the index finger on one side and the ring finger on the other side. Okay, so part of these moved over to the job of responding to the ring finger, part of these were have responded moved over to the job of responding to the index finger. Okay. So you can show that maps are dynamic and they reorganize, and you can explain the experimental results very simply using a map model like this. Now, let us look at a more interesting phenomenon, very intriguing, and which challenged neuroscientists for a long time, and finally the phenomenon found an explanation in terms of very simple map reorganization processes. So, this study is from the book Phantoms in the Brain by V.S. Ramachandran. So, phantom limb is a very intriguing neurological phenomenon. So, people who undergo an amputation of the limb very often have the sensation that the missing limb is still there. They feel that the limb is there, only thing is they cannot see it, but it is as though an invisible missing limb is still present. This is called the phantom limb, and so they feel the hand, and also very often there is a lot of pain associated with this phantom limb. And so it is not just a limb, but this phantom experience is associated with other organs also. For example, there are people who had phantom appendix after appendicitis and an appendectomy when the appendix was removed. So, and they could have phantom nose because the nose is amputated, and so on and so forth, and it poses a clinical problem particularly because of the pain associated with it. There is a long history to the phenomenon of phantom limb. So, for example, Lord Nelson, the famous British general, lost his hand in one of the battles, and he had the experience of sensation of phantom limb, and he thought that it is a direct evidence for existence of soul. Because I mean, it's, if you think about it, you can't see the arm, but you can still feel it as though it is an invisible ghost like presence of the arm is still there. So, he thought that it proves that there is such a thing called soul. Then Philadelphia physician called Silas Weir Mitchell, he coined the term phantom limb, because during the American Civil War, this doctor was treating a lot of war victims, and a lot of these soldiers came with infected arms, and so therefore they had to be amputated, and they experienced phantom limb sensation. There were lots of interesting case studies. So, because this phenomenon was known, but people did not understand what is causing this kind of an experience or a sensation. So, there are a lot of folklore and interesting anecdotes that went with this phenomenon. For example, one story goes like this. So, there was this guy who had this arm amputated, and he went back home after amputation, and he had the sensation of phantom limb, but it did not end there. It became slightly weird beyond that. So, he had this feeling that maggots were gnawing away at his hand. Okay. So, the phantom limb is itself was like weird enough, but on top of that he had this feeling that maggots were eating it away. So, he went back to his doctor who amputated it and complained about this experience. Doctors thought it's crazy, and he just packed him and not doing anything about it. So, after some days, the sensation continued, and then this guy goes back to the doctor, and so then this time doctors takes him seriously, and he had a thought to find out what happened to that arm once it is amputated. So, usually after amputation the arm is sent to the morgue where it is incinerated or burnt, but it so happens that in this particular case, when the arm was sent to the morgue, and the person who is in charge of incineration, he somehow neglected it and then just left it somewhere, and the arm began to rot, and so it was filled with maggots and which were eating it away. And so it's as though the maggots which were eating the dead arm, right, somehow that sensation was felt by this guy who was far away. So I'm sure these are just some cockamamie stories which were made up just to entertain people, and this is because it's totally unscientific and probably not true. Okay, so on a serious note, people have also made up theories to explain phantom limb. One of the earliest theories was just in terms of wishful thinking. So people said that the patients missed the arms so badly, and that created a hallucination of a sensation, a false sensation of the missing arm, and that's all there is to it. Then there were more neurologically oriented theories. One of them explained that when the arm is amputated, the nerve endings, right, are also cut, and at the point of this section, right, they develop what are called neuromas, and where the nerve endings became inflamed and started sending pain signals on their own, and these signals created an illusion of the arm. So this was another theory which is more neurobiologically grounded. Okay, so but interesting turn to this whole study of phantom limbs, or a search for neurological basis or neurobiological basis to the phantom limb phenomenon, came with the work of one doctor at National Institute of Health in the US. His name is Dr. Tim Pons, right. His group, Dr. Pons' group, studied monkeys that have undergone dorsal rhizotomy. So in this is surgery basically where you cut the nerve fibers which carry information from the hand to the spinal cord. So they did that, and then after 11 years after the surgery, when they explored the monkeys again, so they found that the dead hand, that is the hand for which the nerve inputs are cut off, right. When they stroked that hand, there were no neurons in the somatosensory cortex of the monkey to respond to that, okay, which is not surprising because the hand was not sending any signals to the cortex. But what is interesting is when the face of the animal was stroked, right, the cortical regions which are previously responding to the hand, right, to the dead hand, were now responding to the face, to stimulus from the face of the animal, which is a bit strange. Now, Dr. Ramachandran has taken the results from Dr. Pons' studies as a starting point, right, and he had an interesting idea, and he applied this idea on a patient, on a phantom limb patient who visited him. This first patient was called Tom, and he is a 17-year-old young chap. Tom lost his left arm just below the elbow, and so he had the sensation of phantom limb. And what is strange is he could feel that he could wiggle the fingers of the phantom limb, he could reach out and grab objects, and so on and so forth. So, so the experiment goes like this. The doctor has taken a kind of a cotton swab, a Q-tip, right, and touched various points on the surface, the body surface of Tom, and asked him to report which part of his body is being touched. So while the experiment is going on, so this is the patient, and he was actually blindfolded in the experiment. Okay, so when the patient's cheek was touched, and when the patient was asked which part is being touched, the patient reported that the cheek was being touched, and also the missing limb, the missing thumb of the phantom limb. So similarly, when the patient's upper lip was touched, the patient agreed that upper lip is being touched, and also the little finger, right, of the phantom limb was being touched. So similarly, when the lower jaw is touched, the patient reported that the missing little finger was touched. So therefore, stimulation of the face, right, is somehow being combined with the stimulation of the different parts of the phantom limb. Okay, so this simple experiment led to the discovery that there is a whole map of the patient's phantom limb on the face. So for example, here you have the index finger, here you have the thumb, and so on and so forth. So there is a whole map of the hand on the face. Now, in addition to the map of the hand on the face, similar maps were found in two other locations. For example, this whole map on the right shoulder, and when you the other places there is no dual sensation. For example, when you stimulate the chest, there is only experience of touching the chest, but there is no other dual location. In the upper left arm, right, it was found that there is a whole map of the hand. So map of the hand is located in two places. One is on the face here, and another is on the upper shoulder. Now, why is this happening? So, from whatever we have seen about map reorganization, right, how when a part of the map it loses stimulation, so does not receive any stimulation. So, neurons in that part of the map sort of get reorganized and reallocated to responding to neighboring regions. So, and if you look at the somatosensory map, so we know that so this is the hand region, and here you have the shoulder, okay, and so here you have the face. So, the neurons in the hand region do not get any input, right, because the nerves are cut. So, therefore, the shoulder region now kind of encroaches into this the hand region, and the face region also now encroaches into the hand region. This is the reason why there is a whole map of the hand on the face and also on the upper shoulder. Right. So, the somatosensory map region, like I just explained, is flanked on one side by the face area and other side by the upper arm and the shoulder area. So, therefore, when the area corresponding to the forearm does not receive any signals, the neighboring area just expanded and kind of encroached into this area. So, so that gives some explanation of why there is a phantom limb sensation, but there is a real problem. The clinical problem associated with the phantom limb is the phantom pain. So, there were several case studies which Dr. Ramchandran has looked at and reported in his book. So, one of them is a patient called Mirabel Kumar. She was born without arms, and her phantom arms are about 6 to 8 inches shorter than normal arms, and there is no movement of of the phantom limb during walking, but when the patient gesticulates, right, during talking, the arm, right, the arm moves. So, the arm can gesticulate, the phantom limb can gesticulate. Then the other patient called John McGarth, right, and this guy in the phantom limb can also throw the ball when the patient serves in tennis, and during reaching movement. So, it feels as if the phantom limb is zooming out, that is like the target which you are trying to grasp is too far away, it is as though the phantom limb can stretch itself out, right. But what is interesting is in one interaction with the patient, right, the doctor asks him to pick up a cup with his phantom limb, the cup is on the table, and just when the patient is trying to grab the cup, the doctor pulled the cup away, and in that process I do not know, the patient must have felt an extra stretching of his phantom limb, and that created a lot of pain. So, that grabbing was felt to be painful. So, the again and again the phantom limb patients report pain in the phantom limb. So, the question is how do you treat it? So, this is a pretty strange situation, because the phantom sensation itself is is odd enough, right, and how do you treat the phantom pain? Okay, so, to understand why they are feeling the phantom pain, right, here is the theory proposed by Dr. Ramchandran. So, it goes something like this. So, if you look at this this kind of schematic. So, we know that motor cortex originates the motor commands, and the motor commands go to the spinal cord here and way down, and then spinal cord sends commands to the muscles of the arm, and then arm moves. So, at the same time it turns out that the motor cortex sends copies of its command to other parts of the brain. So, we have seen earlier that it sends a copy to the cerebellum, for example. It also sends a copy to the parietal cortex, right, and now this is called a reafferent copy of the motor command. Then parietal cortex also receives sensory feedback. So, it receives input visual and somatosensory cortex. So, visual information tells you where the arm is located and how it looks, its appearance. The somatosensory feedback gives you information about the arm configuration, that is what are the joint angles of the fingers and of the wrist and so on and so forth. So, using that also the parietal cortex can judge where the arm is and with respect to the body. So, now when the parietal cortex receives the copy of the motor command, right, it kind of calculates what would be the expected position of the arm when this motor command is executed. Okay, so, this is what is called a forward model. So, the parietal cortex computes a forward model and estimates where the arm would be, and it also receives the sensory feedback from the visual and somatosensory and other sources. So, it compares its estimated position of the arm with the actual position, and if there is a mismatch between these two, there is an error, and this error is fed back right to the motor cortex, and so asking the motor cortex to adjust its command slightly, so that this mismatch is cleared and the actual position of the arm and the measured position the expected position of the arm, right, are both the same. Okay, so this is what happens in a normal person, but the problem is in case of phantom limb patient, right, there is no visual feedback because the arm is not there, and there is no somatosensory feedback because again the arm is not there. So, therefore, what can happen in principle is the parietal cortex receives the motor command, right, and estimates where the arm should be, but it does not receive any echoing feedback from the sensory organs. So, it thinks that okay, there is a mismatch, the arm is not able to reach the target location, right, and therefore, it sends an error correction back to the motor cortex, asking it to strengthen the give even stronger command to arm, so that the arm can move to the desired location. But this loop will never end because there is no supportive or confirming feedback from the visual and somatosensory sources. So, therefore, this goes on, and it is as though this motor cortex and parietal cortical loop is stuck in a kind of a frozen state, and which probably is translated into some kind of a clenching of the arm, and which is then associated with phantom limb pain. So, Dr. Ramchandran suggested a very simple experiment to relieve this situation and relieve the pain. This is called a mirror box experiment. In this experiment, there is a box and which has two compartments. The two compartments are separated by a mirror, which you can see here, right. And so there are two slots to the box into which you can the patient or subject can put the two arms. When a phantom limb subject is using the box, which is typically the case, right, so in one of the slots the intact arm goes in the other slot there is nothing, because here in this case the right arm of the subject is missing, that is amputated. So, nothing goes into the right slot, only the left arm, the intact arm goes into the left slot, and then the mirror is placed so that it faces the intact arm. So, in this kind of a configuration, if you look at it, it looks as if the patient's right hand is still visible, it is still there, right. So, the patient gets a feeling that both arms are present, right. So, in this kind of a situation, so what happens is the patient can see his left arm because it is actually there, and patient gets sensation that he is seeing the right arm because it is only the mirror image of the left arm. So, visual feedback is restored, although somatosensory feedback is still not there. So, because of this, when the patient practiced on this box again and again, something interesting happened. So, so this was first tried on this patient called Philip Martinez, right, who suffered from brachial avulsion. So, this brachial plexus is basically a kind of a bundle, a kind of a junction area which connects the spinal cord with the arm, and in a motorcycle accident the nerves were kind of yanked out of their nerve roots, so therefore, the patient had a phantom limb sensation. So, when the patient worked with the box after a few weeks, the phantom limb was completely gone. Since the limb itself was gone, the pain also left along with it. Okay, so, what happened in this case, like I just explained, is visual feedback is restored, but the somatosensory feedback is still not restored because there is no real physical arm. So, there are no touch and proprioceptive information coming from the phantom limb. So, probably brain got confused because it is getting mixed signals, because visually it looks like the arm is there, but in terms of touch there is nothing there. So, this conflict maybe created a problem, and it kind of destroyed the illusion of the phantom limb in the process. So, similar experiment was done also in another patient, Robert Townsend. He lost his arm, he lost left arm 6 inches above elbow due to cancer, and he had this kind of the same clenching spasm and the pain along with it. So, when he worked on the mirror box, right, he also got rid of his phantom limb and also his pain. So, now let us look at an interesting aspect of phantom limb and also the question of experience and how neural activity is related to experience. So, there is a problem called the binding problem, so which basically states that if two sets of neurons or two pools of neurons respond to two different objects. So, in this simple schematic, so there is a woman in this scene and there is also a cat, and you assume that different neurons respond to the woman and the cat, and neurons which respond to the cat, right, they all fire in synchrony. So, as you can see right here, and neurons which respond to the woman, right, also fire in synchrony, and neurons responding to the woman and neurons responding to the cat, they are out of sync. Okay, so these and these are out of sync, but neurons which respond to the same object, they fire in sync. Okay, so basically this is just a schematic, but these kinds of schematics have their roots in experimental data. Some of the earliest work on this was done by Gray and Singer on cat's visual cortex. So, where they found that when a moving bar is shown to the cat, neurons which respond to the bar, right, all fire in great synchrony. But when two bars are shown and two bars move in opposite directions, neurons which respond to one of the bars fire in synchrony, neurons which respond to other bar also fire in synchrony among themselves, but there is no synchrony between these two pools of neurons. Okay, so this is called a temporal binding. So, basically you bind all the neurons which respond to a given object together by allowing them to fire in synchrony. Okay, so this way you are using the time dimension to express the identity of a given object, or identity, or you combine all the features of a given object, right, into one identity of an object through the time dimension. So, a classical way of achieving this kind of a binding is by having separate neurons that respond to different combinations of features, as you see on the left side of the slide. Whereas in synchronous firing, so if you look at the example, so there is a yellow triangle and a blue square. So, here you have two color properties, yellow and blue, and two shape properties, that is triangle and square. So, imagine that there are separate pools of neurons which respond to each of these features. So, there are these triangle neurons which see whenever they see a triangle, there is square neurons which respond to a square, and then there is yellow neurons and blue neurons. Right. When you are looking at a yellow triangle, neurons which respond to yellow and neurons which respond to the triangle, right, will be firing in synchrony, right, which is what you see here. Similarly, when since you are looking at a blue square here, neurons which respond to blue, which are these neurons, neurons which respond to the square, which are these neurons. So, they are in synchrony. Okay, so this is called temporal binding, and there is a lot of evidence for this kind of binding occurring in the real brain. So, because of this binding, so when two neurons fire in sync to a given two different features, brain tends to believe that these two features come from the same object. So, this this feature of the brain has been exploited, right, to create very interesting illusions. So, one of them is called the rubber hand illusion. So, in this illusion there is a subject, right, and he places both his hands on the table, and there is a separator, right, which will ensure that the subject cannot see his left hand. But in place of the left hand which is invisible to the subject, they place a rubber hand which kind of looks like his left hand, right, and then the experimenter stimulates the left hand of the subject, right, and the rubber hand, right, simultaneously. So, for example, he has taken brushes and is stimulating the index finger of the actual left hand and the index finger of the rubber hand simultaneously, right, in a correlated fashion. That means the pattern of stimulation on these two fingers is exactly correlated. It should not be strictly periodic, it should be slightly irregular, but the rhythm should be the same, right, on these two points. And when they did that for maybe a few, maybe 10 minutes or so, right, the subject had the feeling that the rubber hand actually belonged to him. Right, he had the feeling that when this stimulation is going on the rubber hand, he felt that his real hand is actually being stimulated. In fact, in one dramatic variation of the experiment, the experimenter tried to hit the rubber hand with a hammer, right, and the patient was so shocked, he jumped up in alarm when that happened, because he was so deeply identified with the rubber hand because of the way in which the stimulation was given to the subject's brain. So, that gives rise to a very interesting philosophical angle to this whole idea of temporal binding. So, what it looks like that what we experience is not really the world, but we only experience our brain's activity, right. To the extent that the world produces a certain reliable response in the brain, right, we experience the world in a reliable fashion, but by artificial manipulation, if you can manipulate the activity in the brain, right, then you can change the experience. So, that gives rise to a whole deep philosophical question of what is really out there. Okay, so, we will stop at this point. So, to summarize, In this lecture, we have seen that the different kinds of brain maps, and brain maps are governed by two important self-organizing mechanisms, and with that we were able to explain several kinds of maps, visual maps, somatosensory maps, auditory maps, and finally we also looked at the phantom limb phenomenon. Thank you.